Assalamu alaikum everyone. Inshallah you are all doing well. Okay, so this is I'm going to do a small presentation for our lesson 1. You have already received the slides in your Google Classroom, and I already give you the slides before. So this is what we will be covering in this lesson. So we will use the Punnett square to predict the result of monohybrid crosses, and we are going to describe and explain the use of test crosses. Okay, so first I'm going to talk about single factor inheritance, and I think this one we are already explained in the school. So for example, here we are following the eyes color. So here, for example, the father is a brown color eye and the mother is a blue color. So the child will inherit from both parents. And let's say this is true breeding. What does it mean, true breeding? This one we already explained, right? So you have here, means actually the father is homozygote for brown color, and the mother is homozygote for blue color. So the daughter and the brown is dominant over blue. So the daughter, she will receive both genes from father and from mother. So this is actually what I have explained here. So let's say the black or the brown, so the brown is dominant. The blue is recessive. Okay, so the black will be we will put the symbol for brown as a B capital and blue for small b. Okay, so okay, so we said breeding true is actually what? Breeding true is actually being homozygote for a specific gene. For example, here we have the flowers, which are pink. So when you cross them together, they will produce only pink color. Then you cross the F1 together, will produce only pink color. So a phenotype for a simply inherited trait is said breeding true if two parents with that phenotype produce offspring of that same phenotype. So if you breed pink color, give you always pink color. If you breed white color, give you always white. Or if you breed red color flowers, will give you always red. So here example, let's say the parents, we can go back to the example of parents. So here you have a breeding true parents. So this one for brown color and this one for blue color. So if you draw the Punnett square, so how is it going to be? The Punnett square will be as follow. So I think you'll see. So this is the father. The gametes will be, so the father is actually capital B because he is brown color, and the brown is the dominant, and the mother is small b, which means that she has blue colors, she has the recessive one. So the father, the gametes he will produce is actually capital B. So this is the sperm produced by the father, and the mother, she will produce eggs which small b. If this sperm fertilizes this egg, you will have capital B, small b. Here capital B, small b, capital B, small b, capital B, small b. So all their children will have genotype capital B, small b, 100%. 100% of them, they will be capital B, small b. And what is the phenotype? What is the feature you will see? Is actually brown color. So this one will be actually the phenotype. and it will be brown color. All of them will be 100% brown color. Okay, so this one you can feel actually the Punnett square. So here, if the parents are capital B, a true breeding, all the offspring will be capital B. So 100% of gametes will be what? 100% of the gametes will be capital B. If the parents are small b, this is the case of the mother, all offspring will be small b. So 100% of the gametes, so the mother will produce which type of gamete? Only small b. So if the parents are capital B, small b, so they are not a true breeding, So in this case, the gametes will be 50% capital B and 50% small b. So here the definition for genotype and phenotype. So phenotype is actually the feature of an organism. That's what you can see. So, but the genotype is actually the genetic makeup, means the gene that person or that organism inherit from his parents. So it's the genetic makeup of an organism in terms of allele present. Like the case here. This person, genotype is small b. This is the case of the mother, isn't it? And this one is capital B. This is the case of the father we have seen just now. So the phenotype is brown, genotype is capital B. Here phenotype is blue, but genotype is small b. And the offspring here, what we call F1. F1 means genotype. F1. Offspring, the offspring. So all the offspring, their genotype is capital B, small b. And their phenotype is brown, because brown is dominant. Okay, so the capital B is called homozygote dominant, small b homozygote recessive, and capital B small b is actually heterozygote because it doesn't have the same genes. So homozygote means having two identical alleles of a particular gene. In this case here, capital B, the gene for the eye color. So, and this one, homozygote can be for both, for dominant and for recessive. So here you can see homozygote dominant, homozygote recessive. And heterozygote means having two different alleles of a particular gene. So here, capital B, small b. And I already explained what is the different allele. It's actually a different version of a gene. So here you have again these two marker pen, but this one is different from this one in terms of color only. So in the genes, in terms of, so I write it here. So in the term of genes, so you have two genes, both for eye color, for example, eye color. So this one will be capital B and this one will be small b. What will be the difference? It will be a small sequence, for example, here. Instead of having an A here, you have a C, or you have a T, or you have a G. Okay, so, okay, now we are going to see the ratio 3:1. This is what we call the ratio 3:1. So let's say here you have the example of a mice which is black, and in this case black is dominant, which is capital B, and white is brown, because I couldn't find any picture which is brown, so I put here a white mouse, and brown mouse is recessive. So that's why this one will be written, this one capital B, this one small b. Okay, so in F1, when you cross these two mice, this one is homozygote, so it's true breeding. This one is homozygote, it's true breeding, and you are going to cross them. So what are you going to have? So let's say we put this one here. So what are we going to have here? Capital B, and this one we will have small b, small b. So here this is F1. Okay, so you are going to have here capital B, small b for all. Capital B, small b, capital B, small b, capital B, small b. So what will be the genotype? Genotype is capital B, small b, 100% of them. All right. And phenotype will be what? Which color is this? Capital B is black. And also 100 of them. 100% of them will be black. So this one, capital B, means will be black, 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 black. Okay. So this one is for F1. Now, because mice, we can cross them together. So you cross another mouse here, which is capital B, small b, which is heterozygote. You cross another mice with this one, means two black heterozygote. So what are we going to have? So this is F1, and now you cross F1 with another F1. So this is the one. So capital B, small b crossed with another F1. So these two are from F1. So F1 crossed with F1, you will get what we call F2. So what are we going to have here? What are the gametes made by this one? So this one will make the gametes capital B, small b. This one will make the gametes capital B, small b. So what are we going to have here? Capital B, capital B, capital B, small b. Here, capital B, small b, and small b, small b. Okay, so now if we talk about genotype, what are we having? We will have capital B, capital B. We will have capital B, small b, and we have small b, small b. What is the ratio? So this one is 25%, this one is 50%, and this one is 25%. Okay, now this ratio we can write it 1 to 1. Okay, so 1 to 1. This is for genotype. What about phenotype? Phenotype will be, this one will be black, this one will be black, this one will be black. So we have black, and we have another phenotype. This one will be, remember the parent one, this one will be brown. So how many percent black we have? 1, 2, 3. We have 75%. And brown we have 25%. So means we can write it in ratio 3 to 1. So this is the ratio 3 to 1. This is what it means. The ratio 3 to 1. So the 3 to 1 ratio. And you have the example here of the flower. Again here, so you are going to cross white flower with pink flower or purple. So white flower is recessive, purple is dominant. So purple is dominant, so we will write it as capital P, because this one is true breeding. Then white we will be written as small p. I will put a line so you can know that this is small p. So in F1 generation, if we cross this one with this one, what are we going to have? So the gametes made by this one will be capital P. Gamete made by this one will be small p. So here will be capital P, small p, capital P, small p, capital P, small p, capital P, small p. So what is the genotype here? all capital P, small p. What is phenotype? Will be, they all have capital P, so what will be the color? It will be purple. Purple, 100% of them. Here also 100%, no? You have to write the ratio. Okay, now if you cross F1 with F1, so this is what I have here. Now you cross F1 with F1. So F1 with F1, so this one is capital P, small p, capital P, small p. If you cross them, you draw the Punnett square and you will find the ratio. So this one will be capital P, small p, capital P, small p. Here you will have capital P, capital P, capital P, small p, capital P, small p, small p, small p. Okay. If we write genotype, what do we have? You have capital P, capital P. We have capital P, small p. And we have small p, small p. What is the ratio? This one, 25% means 1. This one, 2. And this one, 1. Means 1, 2, 1. What about phenotype? So phenotype, this one got capital P, purple, capital P, purple, capital P, purple. So we will have purple. And this one, small p, will be which color? Will be the white. So this one will be how many? How many purple? 1, 2, 3. So 3, 2, 1 white. So you have the ratio 3 to 1. Okay, so this is actually what I wrote here, what is mentioned here. So this is the ratio 3 to 1. Okay, so this is actually summarizing the whole thing. So we have here 3 purples, 1 white. So this is phenotype, the one you can see. And genotype is actually the genes or the alleles. So what do we have here? Capital P, we have 1. Capital P, small p, we have 2. Remember, we have 2. This one and this one. And homozygote, you have 1. So the ratio for genotype is 1 to 1. The ratio for phenotype is 3 to 1. Okay, and the last thing I want to discuss here is the what we call test cross. What is a test cross? A test cross is actually is a test we do when you don't know the genotype of an organism. So usually which one we don't know. Let's say the case here, you have a purple flower. This purple flower can be homozygote, capital P. So this one will be purple. Or it can be capital P, small p. So if you want to know exactly what is the genotype of this flower, you are interested to know. So how are you going to do it? It's actually very simple. So we know that white is recessive. So what is the genotype of recessive? So it will be small p. This one we know already. So what we are going to do is to cross the flower which we don't know, unknown one, with the one which is recessive, which we know the genotype already. So in this case here, you will have 2 scenarios. Let's say the first scenario, your flower is capital P. Okay, this is the first scenario. You are going to cross it with small p. So if you draw the Punnett square, what are you going to have? So this one, the gametes will be capital P. This one, the gametes will be small p. What are you going to have here? You will have capital P, small p, capital P, small p, capital P, small p, capital P, small p. And this is what we call F1. Means if you cross your unknown flower with this recessive one, and you got all the flowers here, which color? All they have capital P, means they are purple. 100% of your flowers are purple. So in this case, your genotype, your flower, it's actually capital P, capital P. Okay, so now the second scenario. If your plant is capital P, small p, so I am going to predict what I am going to have. So if your flower is capital P, small p, you will cross it with small p, small p. Your flower is heterozygote, so what are you going to have here? Capital P and here small p. You are going to cross it with small p, with the recessive one. What are you going to have here? Capital P, small p, capital P, small p. Here, small p, small p, small p, small p. So what do I have here? This one will be purple. What will be the percentage? 50%. And this will be white. And the percentage will be also 50%. Okay. So this is actually in theory what you will get. So if you do the crossing of your unknown flower with the white and you get 50% white, 50% purple, means your plant is heterozygote. If you do the crossing of your plant and you get 100% purple, means your flower is homozygote dominant. So this is actually the case, sorry, this is actually the case here. This is what I explained here. So this is why we need a test cross. So how can we tell the genotype of an individual with the dominant phenotype? Such individual could be either homozygote dominant or heterozygous. So the answer is to carry out test cross. So this is what we call test cross. Means breeding the mystery individual, the one which we don't know, with the homozygote recessive. Because the recessive, you know what is the genotype. So if an offspring display the recessive phenotype, the mystery parent must be heterozygote. And you have the example here in this. So you have the example. This one I remove. So you have the example here. So you have a purple flower. You don't know the phenotype, the genotype. And here you are going to cross it with a white one, because the white one we already know. So what are you going to have here? This is the first scenario. I put here one. This is number 1 scenario. And this is number 2 scenario. So if you cross it and you got all of them purple, means your plant here is a homozygote dominant. If you cross it and you got half purple and half white, so your plant is heterozygote. Okay, I want you to do this one. So let's do it. Let's say here I have a black dog, and I know that the black is dominant, the brown is recessive. So how am I going to do? I am going to cross my dog, which is mystery dog. I don't know this dog, is it can be capital B, sorry here, it can be capital B, or it can be capital B, small b. So if we want to know, how are we going to do? We are going to cross it with the recessive one. The recessive one is a brown dog. So I got here a brown dog. So my dog here is what? For sure it's small b, small b. Okay, I will first scenario. I will do the first scenario, and later you have the second scenario. So here I will put my dog here. Let's say my dog, this one is capital B, capital B. So the gametes will be capital B, and this one will be small b, small b. So here capital B, small b, capital B, small b, capital B, small b, capital B, small b. So here all the offspring will be which color? They will be all black. So if the second scenario is this dog is actually capital B, or I put it here, sorry, I put it here, capital B, small b, heterozygote. Okay, the gametes will be capital B, small b, and here this one, the gametes will be small b, small b. Okay, so here if I cross this, capital B, small b, small b, small b, and here I will have capital B, small b, small b, small b. Okay, so this is what I will get. Means how many will be, so this one will be, this will be black. So means I will have black dogs, and this will be brown, like the mother. Okay, what I need to do, okay, now I have in theory what I need to do. So now I just cross these two dogs and wait. See, if all the dogs are black, means the father here is homozygote dominant. If I got 50% black, 50% brown, means the dog, the father here is heterozygote. Okay, so you just do your test and see. Let's say we got this one. We got half of the dogs puppies, half of the puppies brown and half of the puppies black, means the father is actually heterozygote. But if all the puppies are black, means the father is homozygote dominant. And the end. So that's all. I hope everybody understand. So see you the next lesson, inshallah.